Now that you're familiar with the induction and the general aspects of neurocrest cell migration, we're going to look at the migration and the differentiation of neurocrest cells from different regions of the embryo, beginning with the trunk neurocrest cells. Now, these cells take two different migratory pathways. They can migrate ventrally or dorsolaterally. The cells that migrate ventrally migrate through a portion of mesoderm that's known as the sclerotome. And the sclerotome is simply a portion of the somite that's going to give rise to the cartilage of the spine. So the cells that take the ventral pathway migrate through the anterior portion of each sclerotome, and they give rise to neurons and glia of the peripheral nervous system. Now, the other cells of the trunk migrate through the dorsolateral pathway, and these cells give rise to melanocytes, which are pigment cells. And their migratory path is such that they're actually migrating between the future epidermis, which is the ectoderm, and the future dermis, which is formed by the mesoderm underneath it. So they eventually colonize the epidermis by entering through small holes of the ECM, the extracellular matrix of the epidermis. Now, just so you can orient yourself as to what we were looking at before, the neural crest cells are going to be delaminating from the dorsal aspect of the neural tube. And the trunk cells, the ones that migrate through the ventral pathway, are going to go through this portion of the mesoderm, which is the somites. And the somites have at this region mesenchymal cells. And you can see this here. This is the dermomyotome that is going to give rise to the dermis and muscle. And the sclerotome through which the ventral cells migrate is here. This is what gives rise to the cartilage of the spine. And so the cells that take the ventral pathway will migrate through this aggregates of mesenchymal cells that make up the sclerotome, specifically through the anterior portions of the sclerotome, because they have both an anterior and a posterior portion. The first group of neurocrest cells that leaves the neural tube in the trunk region enters to the ventral pathway. And the reason for that is because they are blocked from entering the dorsolateral pathway by ECM molecules that are secreted here and by juxtacrine factors. So these factors keep these early cells from entering the dorsolateral pathway so that they enter the ventral pathway instead. In addition to that, the cells initially are able to pass through this region that's located between the somites, this space that you see here. But eventually that space also becomes populated with extracellular matrix molecules and factors that prevent the cells from entering that space. And so eventually the ventrally migrating cells can only pass through the anterior portion of each sclerotome. So these factors that prevent the neurocrest cells of the ventral pathway from entering into the dorsolateral pathway and that prevent them eventually from migrating between the somites include molecules of the ECM, secreted proteins, and also juxtacrine factors. Now, as I just mentioned, these cells that take the ventral pathway migrate only through the anterior portion of each somite, specifically through the anterior portion of the sclerotome of the somite. But they cannot migrate through the posterior portion, so here they get stuck. And the reason for that is because in the posterior portion of the somite, there are molecules that prevent the migration of these cells. So there are juxtacrine factors like ephrins and secreted factors like the factor semaphorin 3F that prevent these cells from entering the posterior region of each somite. At the same time, in the anterior portion of the somite, you have extracellular matrix molecules that promote the migration of these ventrally migrating trunk neurocrest cells. If we were to label the juxtacrine factor ephrin, we would see that its expression, which is in the posterior portion of each sclerotome or each somite, does not correspond to the location of neurocrest cells. And this pattern of neurocrest cells is important because it is needed to preserve the patterning of the nervous system. Remember that these neurocrest cells that enter into the ventral pathway, some of them give rise to dorsal root ganglia, which are sensory ganglia that bring sensory information into the spinal cord. In mice, it's been shown that knocking out genes like semaphorin 3F and ephrin disrupts the patterning of the dorsal root ganglia, which are the sensory neurons. And you can see that in the wild type for semaphorin, the neural crest cells, which are shown in the purple, enter only through the anterior portion of the somite, but not the posterior portion. But when semaphorin 3F is knocked out, as it's shown here in panels G and H, you can see that the neural crest cells can enter both through the anterior and the posterior compartment of the somite, and that disrupts the dorsal root ganglia patterning, as you can see here.